Hello, everyone. I'm Qiang Song, and a scientist of neurobiology in RD department at GemPharmatech company. And today, I'm very glad to introduce our preclinical mouse model for neurodegenerative disease. And I would like to give a introduction to our AD and PD mouse models. Today, my presentation can divide the 4 parts. And firstly, I would give a brief introduction about the neurodegenerative disease. And second, we will introduce the AD mouse model called the FADT mice. And then third part of my presentation, I would like to introduce 2 Parkinson disease mouse model. And at last, I would like to give an introduction about our platform for neuroscience research. And first part, I would like to give some brief introduction about the neurodegenerative disease. As we all know, the neurodegenerative disease are a major cause of disability and premature death among all the people worldwide. The clinical manifestation of a particle neurodegenerative disease can reflect the region of the brain, and the specific population of cells can be affected. And in the Alzheimer's disease, the pyramidal neurons in the LC, EC, and hippocampal regions can be affected and form the A-beta plancot deposition. And in the Parkinson disease, the dopaminergic neurons in the substantia nigra can also detect the alpha-synuclein protein accumulation in the cells. And another different type of the neurodegenerative disease, like the Huntington disease and the ALS, also have the similar phenotype performance like the PD, AD, and also some different type of the neurodegenerative disease models. And there are 3 models have been proposed in neurodegeneration, although the beginning and the end of the process are similar in the 3 hypotheses. And the events that induce the neural death are very different. And firstly, we can show this in the loss of function hypothesis. There may lack of the activity of the native proteins is a key step which initiate the death of the neurons to cause the neurodegeneration. And in the gain of toxicity activity hypothesis, the crucial process is neurotoxicity to cause by the misfolded and aggregated proteins. And in the inflammation models, neural death is indirectly mediated by the supported cells like the astrocytes and the microglial cells. And in some disease, a combination of those mechanisms might cooperate. And the hallmark events in neurodegenerative disease is misfolding, aggregation, and accumulation of proteins, which lead into the cellular dysfunction and lost the synaptic connections in the brain, and eventually to cause the brain damage. And despite the involvement of distinct protein in the different neurodegenerative disease, like in the AD pathology, have shown that the A-beta deposition and they have the tau inclusion in the some type of dementia disease, and also the alpha-synuclein inclusion in the Parkinson disease also can be detected, and some type of TDP43 inclusion in the ALS and FTLD FTLD disease. And also this disease can be occur in the part of the brain region and can separate it in whole brain at the late age of the neurodegenerative disease phase. And this process of the protein misfolding and aggregation is remarkably similar. And the misfolded protein can aggregate and also can self-propagation of the pathological protein aggregates in the neurodegenerative disease. And besides the protein seeding and the propagation, there are some common neuronal pathways can operate in the multiple neurodegenerative disease. which including the protein quality control and the autophagy lysosome pathway changes, and also some type of the neurodegenerative disease can affect the mitochondria homeostasis and also induce the synaptic toxicity and cause the neuronal dysfunction. And the mouse model are a powerful tool to mimic our human disease. And the GPT has generated different type of mouse models based on the gene editing technologies, which including the knock-in, transgene, and knockout as the humanize the mouse model to mimic the human disease or import the drug target gene in the mouse models. And this slide have been shown that the neurodegenerative disease mouse models at the GPT company. And today I would like to give a brief introduction of the highlights in the red colors show that the FADT mice and another two type of Parkinson disease mice called the SNC A53T mutation point mutation mice. Another type is the T5CKO mice. And there is a table to show that another different neurodegenerative disease mouse model at the GPT, and the timeline you can available in our company. And firstly, in the second part, I would like to give a brief introduction about the AD mouse models called the FAD40 mice. And in 1907, the 2K histopathological hair markers of Alzheimer's disease, like the amyloid beta plankoid deposition and the neurofibrous plankoid formation in the AD patients, can be detected by the Dr. Elias Elmos, and which can cause the brain size decrease in the Alzheimer's disease patients. And there is research published in this year. They show that the data have shown that estimated 6.5 million Americans aged 65 and older are living with Alzheimer's dementia in 2022, and about 73% are aged 75 or older. And also the women live longer than men on average. So the older age is also a great risk factor for the Alzheimer's disease. And we can see that the women patients have the higher frequency compared to the men groups. And also the cost of the healthy care and the long-term care for individuals with Alzheimer's or other dementia are substantial. And the dementia is one of the closest condition to society, and it have been reported that till 2015, there are about a lot of people have the dementia phenotype. So the dementia is a very huge market to our studies of industrial drug development. And Alzheimer's disease is one of the most popular dementia disease in the world. So the AD drug development is a hot research field in scientific research and industrial. And this slide have shown that list the different clinical phase of AD drugs, and there are about 143 agents. Carry out 172 clinical trials until 2022 years. And these clinical agents are based on the different AD pathologic mechanism, which included the amyloid mechanism and the tau pathology, and also target the inflammation. Also, some of them to target the synaptic plasticity or to function as the neuroprotection and so on. And although there are many preclinical agents, but still less FDA-approved drugs. Until now, only 7 drugs have been approved by FDA, and 4 of them is the acetylcholine inhibitor, which is developed based on the cleavage hypothesis. And one of these, the NMD receptor agonist based on the excitatory amylo acid neurotoxicity hypothesis in Alzheimer's disease. And in the last years, there are antibody drugs called aducanumab. Is the anti-A-beta nanoclone antibody was conditional approved, raising the confidence of the AD amyloid pathological hypothesis. And until now, there have a lot of GWAS study uses many thousands of samples have identified more than 20 genetic factors. And those genes show the different frequency and the risk factor to AD pathology and have multiple functions involved in the APP, toll metabolism, and also the cholesterol metabolism. Some of them involve in the immune response and to regulate the cytoskeleton development. And to mimic the AD pathology based on amyloid hypothesis in mouse models. And we only focus the APP, PSEN1, and the PSEN2 genes. And those genes are highly related to the amyloid genetic mechanism of Alzheimer's disease. And we all know that mutation in the PSEN1 genes leading to generate different A-beta polypeptide. And the APP mutations can increase the A-beta levels and also increase the ratio of the, some kind of mutation can increase the ratio of the A-beta 42 and the A-beta 40, which is important health markers for AD pathology. And here we develop the FAD40 mice. This is a new models to explore Alzheimer's disease pathology and therapeutic efficiency. And these mice carry 4 AD mutations, Swedish and Indiana mutations in the human APP genes, and another 2 mutations is in the PSEN1 genes. And compared with the 3TG and 5AD mice, which is not available to company and for commercial use. Our FAD40 mice have independent IP, so the cells are not limited. Everyone can use it. And besides that, they'll have a lot of advantage of our FAD40 mice. I will show you later about our preliminary data to deliver the our advantage of our mouse models. And firstly, we detect the A-beta deposition in FAD40 mice brain, and it's clearly to see that the A-beta deposition appear in the 2 mouse FAD mouse models, like in the cortex and the hippocampus, or show this clear of the A-beta deposition in the brain area. And we also quantified the A-beta Planck area in different brain regions in FAD40 mice. And clearly indicates that the A-beta deposition have shown that the age-dependent increase in the FAD40 mice brain. Among all kinds of A-beta isoform, A-beta 14 and A-beta 42 are believed to be the most important one. We quantitative analysis of the A-beta 40 and A-beta 42 in mouse brain also reflects the similar increase pattern at the A-beta deposition in the FAD40 mouse model. And we have the collaborator with our collaborator to show that compared to the 5FAD mice, the A-beta deposition in the different brain region of 4 months old FAD40 mice are comparably to the 5FAD mice. And also, recently study indicated that CNS inflammation has become the most popular health markers in AD research. So we use the GFAP and ABU1 antibody to detect the astrocytes and microglia in the brain regions, mostly in the cortex and the hippocampus regions. This slide shows the representative imaging of 4 months old Y-type and FAD40 mice. and clearly increase the GFAP positive and IBA1 positive cells in the cortex and the hippocampus regions in the FAD40 mice. And we also quantitative analysis the GFAP positive and the IBA1 positive cell density in the cortex and the hippocampus region in Y-type and the FAD40 mice group. So it revealed that the gliolysis in the FAD40 mouse models. And our data also indicates that the glial cell activation in FAD40 mice at early age of 2 months old. So they have no picture have shown this this conclusion, and we have the data to support this conclusion. And we also to validate the neuronal loss in the AD mouse model. And the neuronal loss is a common pathological health markers in the Alzheimer's disease, and we use the new antibody to send the neurons in different age of mouse Y-type and the FAD40 mice brain. And here we observe the loss of the neurons in FAD40 mice brain in 10 months old age in the cortex and the hippocampus regions. And another brain regions also have shown the similar neuronal loss phenotype in the FAD40 mice. And besides that, we also use the Western blot to detect the synaptic protein PSC95 to expression level to quantify the FAD40 mice synaptic loss phenotype. And this data have indicates that the synaptic loss in 10 months old FAD40 mice and have the tendency decrease at 8 months old. And at here, I would like to give a little summary about the FAD40 mice pathological phenotype. And firstly, we detect the A beta pathology initiation in 2 months old FAD40 mice and have shown the age-dependent increase of A beta 14 and A beta 42 in the FAD40 mice. And also, we detect the gliolysis in 4 months old FAD40 mice. And another feature is that the neuronal loss and the synaptic loss at almost 10 months old in FAD40 mice. And to document the behavioral changes in the FAD40 mice, and we employed a battery of behavioral test paradigm in Asian and 6 matched Y-type and FAD40 mice. And here this slide shows the open field test result in the 8 month of age Y-type and FAD40 group. The total travel distance is have no significant decrease between the FAD40 mice compared to the Y-type mice. As the time in the central area is significantly increased in the FAD40 mice. This data have suggest that the cognitive ability may have impaired in the FAD40 mice. And we also set up the Y-maze test to detect the spatial learning and memory changes in FAD40 mice. And here is the statistical results indicates the spatial learning working memory have decreased in the 8 months old female FAD40 mice, but not in the male group. And to better characterize the FAD40 mice cognitive ability changes, the classic experimental paradigm, Morris water maze, was employed to document learning and memory ability changes. And in the training phase, it clearly to show that in the female group, the escape latency is longer than Y-type control group. So it suggests that the FAD40 mice have the learning ability changes compared to the Y-type group, and also have similar tendency changes in the male group. And we also analyzed the 3 different parameters in the proper test phase. And from the data, and we found that the number of crossing platform and the number of entry to the target content and the time is decreased. The number is decreased, and the time for the gas into the target content compared to the wild-type mice, the FAD40 mice have increased their times. And this data have suggest that FAD40 mice have the deficiency in their learning and memory ability, especially in the female group. But in the male group, we haven't detected the significant decrease between these different parameters in the proper test phase. So our conclusion is that our data suggests that the female FAD40 mice have the learning and memory deficiency at 8 months old, but not in the male group. And we also carry out the drug test in the FAD40 mouse model, and we use a different dosage of drug. We call the low dosage, medium, and high dosage. And this group is this four group is all used in the FAD40 mice. And compare in the black lines is the wild-type control. All of the FAD40 group, the escape latency is longer than the wild-type control. But in the proper test phase, it's clearly to show that compared to the wild-type control, the FAD40 group is learning ability is decreased compared to the wild-type control, but low dosage of the drug have observed significant increase their learning ability in the FAD40 mice. So our data suggests that the low dosage of drug can improve the learning and memory ability in FAD40 mice in the Morris-Watermaze test experiment. And there is a summary about our FAD40 mice. We detect the A-beta plancode as around 2 months old, and the gliosis is also detected in about 2.5 months old. And we detect the cognitive impairment almost in the 8 months old. And the synaptic and the neuronal loss also can be detected in the FAD40 mice almost to the 10 months old. And compared to the 5FAD and the 3TG mice, our FAD40 mice have some advantages listed here. And first is the early occurrence of the pathological phenotype, such as the A-beta and the glial cell activation. It's very suitable for customers who conduct research and drug development to target the pathological phenotype. Another advantage is that the cognitive impairment appeal is later than other products. It's also suitable for the drug evolution studies, which can require a longer innovation period. And there are many risk factors that have higher frequency, but less effect to induce Alzheimer's disease. And most of them are found in sporadic AD patients. And to facilitate the drug development of those potential drugs, we humanize these targets and across the humanized mouse model to the FAD40 mice, which not only carry the drug target, but also have the AD pathological phenotype in our AD plus mouse model. And here is a table we already generate the humanized mouse model and the timeline you can access in our company. And in the third part, I would like to give a brief introduction about our Parkinson disease mouse model. And firstly, we give a brief introduction about the Parkinson disease. The Parkinson disease is a progressive neurodegenerative disorder characterized by selectively and progressively degeneration of the dopaminergic neurons in the striatum, in the substantial neuron, and have the clinical features like the trauma and brascular and ridiculous. And the pathological features of the Parkinson disease is the degeneration of the dopaminergic neurons and form the alpha-synuclein protein accumulation in the living bodies. And Here is a milestone in Parkinson's disease research and discovery, and it has a long journey, about 2 centuries of the Parkinson's disease research, and first discovered by the Dr. GemPharmatech, and sequentially, many pathological features of the Parkinson's disease were discovered, like the dopaminergic deficiency and the microgliosis, and also the alpha-synuclein accumulation in the Lewy bodies. And besides that, and not only the genetic risk factor can induce the Parkinson's disease, but also the external substance also can induce a Parkinson-like phenotype. They have reported that MPDP expulsion can add the Parkinson's disease risk factor. And also, researchers have been identified the mutations in the SNCA genes. It have been related to the Parkinson's disease pathology. And the recent study also indicates that alpha-synuclein protein can performance the cell-to-cell transmission, and this raises the potential therapeutic strategy for Parkinson's disease. And the SNCA gene was first identified as the Parkinson risk genes, and the pathogenetic mutations in the SNCA gene can cause the alpha-synuclein protein conformation changes and the protein accumulation in Lewy's body, and which can lead to the disease pathology. And based on the genetic pathogenesis of the Parkinson's disease, we generate a mouse model which carry the A53T point mutations in human SNCA cDNA sequencing to express the mutant SNCA protein in mouse models. And we have detected the phosphorylated SNCA protein in the mouse brain, which indicates that pathological features of Parkinson's disease also suggest this mouse model have been expressed the alpha-synuclein proteins in the mouse brain. And besides that, we also checked the alpha-synuclein protein expression in the median brain, and those data show that the cytoplasmic aggregation of alpha-synuclein protein in the SNCA53T mouse, it starts at the 8-week-old. And besides that, another hallmarker of Parkinson's disease, hallmarkers of Parkinson's disease is the dopaminergic neuron loss. And we detect the dopaminergic neuron loss in the VTA and the SN regions at 60 weeks of age in our SNCA53T mouse models. And we also carry out a battery of the motor behavior tests in the SNCA53T mouse models. We use the graph strength and the root and rod test to document the motor ability of our SNCA53T mouse. And in the graph strength test, it clearly to show that in whatever the female and the male group have decreased their grip strength at 22-week-old, and have the tendency to decrease motor ability at the 18-weeks-old mice. And besides that, we measured the body weight changes in the SNCA53T mouse models. It can show that the reduction of the body weight in the male group, but not in the female group in the this mouse model. And compared to the Jackson SNCA53T mouse model, our mouse model shows the more severe phenotype compared with these mouse models. And besides that, the alpha-synuclein pathogenesis is one of the Parkinson's disease occlusion. And another pathogenesis is the mitochondrial dysfunction. There are a lot of papers have been published that the mutations in the mitochondrial transcription factor A have linked to the Parkinson disease and also have some relation to the Alzheimer's disease. So we would like to wonder whether direct genetic impairment of respiratory channel function in dopaminergic neurons can recapitulate the features of Parkinson disease in mouse model. And to test this hypothesis, we generate T5 flux mice and cross with the dopaminergic specific Cre mice to knock out the T5 gene in the dopaminergic neurons. And these mice we call the T5 CKO mice. And we detect the dopaminergic neurons in the SN regions and the VTA regions of at a 10-week-old. 10 weeks old T5 CKO mice. It's clearly to show that the dopaminergic neuron loss in the SN regions at this time point, and also the VTN regions also lost some of the dopaminergic neurons in 15 weeks old mice. And we also document the body weight changes in the T5 CKO mice. It's clearly to show that the decreased body weight of the T5 CKO female and male group. And these two groups of mice have the similar decrease of their body weight start at 6-week-old. And we also use the motor ability test paradigm to check their motor ability changes in the T5 CKO mice. We use the graph strength and the rotor rod test to document the motor ability changes in the T5 CKO mice. And clearly to show that the graph strength have the progressive decrease in the T5 CKO group start as about 20-week-old. And in the rotor rod test at 8-week-old, 8-week-old T5 CKO mice also have the motor ability deficiency in the T5 CKO mice group. In the female and the male group also have the similar decrease pattern. And at the last part, I would like to give a brief introduction about our platform for neuroscience studies. And we have a comprehensive behavior analysis platform in the motor ability analysis, such as the open field, graph strength, even the gait analysis. We can analyze the gait changes in the different mouse models. And in the learning and the cognitive test, we can use the no object recognition and YMAs and Morris-Watermaid test to test the learning and memory changes. And also we have some mental disorder analysis paradigm. And besides that, we also have the pathological we also provide the pathological analysis service like the histologic analysis, and another is the molecular biological analysis. Also, our platform have the high working throughput. So very thank you for the time for share the preclinical model for neurodegenerative disease in our company. I will stop here and very hope for your question. Okay. The first question is, will you share slides with meeting attendance? Yes, we can share these slides. And the second question is, what's the advantage of our FADT mice? Okay. In our FADT mice, we can see clearly the Alzheimer's disease pathology. such as the A-beta deposition and neural loss, synaptic loss, and the glial cell activation. And besides that, it shows the observed cognitive behavioral changes and the also some advantage I have mentioned in this slide. And another okay. The third question is, did the FAD40 mice have a possible drug test? Yes, it's still ongoing work. I hope we can share the data as soon as possible. And another question is, are you planning to test some traditional drug efficiency on your PD and ALS mouse model? Yes, it's still on the plan. So thanks this question. And there is a question to wonder what kind of drug delivery routes do we have? Yes, we have the usual route to deliver the drug to the central nervous system, such as intravascular injection and therapeutic injection, and the intragastric administration of the drug targets to our mouse model. Okay, there is a question to wonder do we have the data comparing the behavioral test results to 5FAD mice? We only collect the phenotype data in the website of the 5FAD mice. We cannot access the 5FAD mice in our company. So we haven't this part of the data to show the comparison to the 5FAD mice. Okay, if you have no question, I will stop here. Thank you for your attention. Okay, bye-bye.